Okay, Assalamualaikum and selamat sejahtera. So now let's proceed to the process of Meiosis One. Okay, so remember Meiosis One is to separate homologous chromosome. Okay, so before we continue, so you have to remember Meiosis One can be divided into 4 stages. Okay, the first stage are Prophase One, next Metaphase One, then Anaphase One, the last one Telophase One. Okay, so we start with Prophase One first. Okay, so how to explain the process of Prophase One? So actually there are so many terms that you have to remember during Prophase One. Okay, so one of it here we have chiasmata or chiasma, and then it has tetrad, bivalent, synapses, and then you have to differentiate between sister chromatid and also non-sister chromatids. So actually all of this I already explained in the previous video. Okay, so we look one by one. What happened during Prophase One? Okay, so actually Prophase One, it is the longest phase in Meiosis One. Okay, and then what happened to the chromosome in Prophase One? Chromosome condense and become visible. So condense here, you can also write as chromosome will become tightly coiled or shorter and thicken. So you have to take note. Mesti kena ada 2 point dekat sini. So you have to mention chromosome condense or chromosome shorter or become shorter and thicken and become visible. Become visible ni kena ada because of what? Sebelum berlakunya condense of the chromosome, okay, the chromosome or chromatin is invisible. Okay, so and then next, homologous chromosome pair up. So you have to mention here homologous chromosome pair up, or we said that it will undergo synapses. So synapses actually it is the process by which a homologous chromosome is pairing up. Dia mesti kena berada bersebelahan, okay, secara berpasangan. Untuk apa? To form bivalent or tetrad. Okay, so maksudnya this structure we call as tetrad, and then this is also a bivalent. So kalau tetrad ni dia refer kepada a group or a cluster of the 4 chromatids 1, 2, 3, 4. Kalau untuk bivalent, it will refer to the pair of homologous chromosome. Okay. And then next, what happened during Prophase One? Crossing over occur. So if you look at here, crossing over occur in between the non-sister chromatids of homologous chromosome. Okay, chiasmata here we refer to the region where crossing over is occur. So awak tengoklah dia punya tempat persilangan. Okay, so maksudnya tempat persilangan, okay, ataupun kita panggil sebagai the X shape where the crossing over is occur. Okay, so we call that as a chiasmata. So remember what is the importance of the crossing over? Okay, there is an exchange of the genetic material between homologous chromosome. Okay, so beside, okay, what is the importance of the crossing over? Actually, it will increase the variation, okay, for the daughter cell lah. Maksudnya bila berlaku exchange of the genetic material, the daughter cell tu nanti dia akan dapat genetic recombination between the maternal chromosome and also the paternal chromosome. Okay, so apa yang saya tunjukkan dekat sini adalah contoh bila sahaja parent cell dia adalah 2n equal to 4. So means that you have to draw 4 chromosome. Okay, write this. And then maksudnya dekat sini dia akan ada 2 pairs of the homologous chromosome. Dan satu lagi macam yang saya katakan sebelum ini, bezakan kromosom dari segi warna. Okay, and then next what happened in this stage also, spindle fiber starts to form. So this is the spindle fiber, and then the centrosome migrates to opposite pole. Okay, so actually here the process is same, okay, with the mitosis. Okay, and then nucleoli and nuclear envelope disintegrate. Okay, and then next, microtubule from one pole, okay, or the other attached to kinetochore, one at the centromere of each homolog. So if you look at this one, macam yang ni lah kita akan nampak, okay, there is a microtubule that attached to the centromere, okay, at each of the chromosome here, at each of the homologous chromosome. Okay, and then what happened after that? Microtubule move the homologous pair like this, okay, towards the metaphase plate. Because of what? After Prophase One, okay, the stages will proceed with Metaphase One. Okay, so now we look at Metaphase One. So what happened during Metaphase One? Okay, firstly, if you want to draw the stages of the Metaphase One, so make sure you draw like this. Means that, okay, the chromosome, okay, the structure of the chromosome, homologous chromosome, you have to draw side by side like this. Okay, and then there is a X shape here lah, tempat persilangan. Maksudnya kita akan tunjukkan bahawa kromosom tu bersilang lah in between this homologous chromosome. And then microtubule attached to only one side of chromosome. Okay, so this microtubule dia akan attach kepada only one side sahaja. Di tengah-tengah di antara this chromosome, red color chromosome and also the blue color chromosome, there is no microtubule ataupun spindle fiber. So jangan letaklah spindle fiber di tengah-tengah sini. Okay, yang ni adalah microtubule that attach to the centromere of this chromosome. Okay, and then if you look at the events, okay, kita tengok dulu the chromosomal behavior during metaphase 1. So what happened? Pair of homologous chromosome align independently on the metaphase plate. Okay, so what happened to this homologous chromosome here? Okay, lepas tu satu lagi saya nak ingatkan awak, since crossing over is already occur, so you have to show. Means that, okay, a portion of this blue color chromosome dia dah dapat dah color merah dekat sini. Yang ni pun, yang the red color here dia dah dapat the genes from the blue color chromosome. Okay, as well as this one lah. So awak mesti kena tunjukkan. Okay, and then the important event that occurs during metaphase 1, so you have to remember pair of homologous chromosome align, okay, independently on the metaphase plate, and then one chromosome in each pair facing each pole. Okay, so like this lah. Okay, and then both chromatids of one homolog are attached to microtubule from one pole. So means that di bahagian sini, okay, so this microtubule dia akan attach kepada the centromere for each of this chromosome. Okay, and then the chromatids of other homolog are attached to microtubule from the opposite pole. So untuk bahagian opposite pole tu pun sama, mesti akan ada attachment of the microtubule at the centromere. Okay, and then satu lagi saya nak ingatkan how to draw the microtubule or how to draw the spindle fiber. Okay, so let's say if there is no attachment of the spindle fiber to any of the chromosome here, so make sure your spindle fiber must be overlapping like this. Okay, and then this is the metaphase plate. Okay, now we look at the next stage, which are Anaphase One. Okay, so what happened during Anaphase One? Pair of homologous chromosomes separate and move to opposite poles. So if you look at here, so this homologous chromosome, this pair of the homologous chromosome will separate, okay, and it will move to the opposite pole like this. Okay, and the spindle fiber pull homologous chromosome. So the spindle fiber that attach to the centromere at each of the homologous chromosome, it will pull, okay, the homologous chromosome towards the opposite pole. And then each chromosome still has two chromatids attached at their centromere. So perbezaan di antara Anaphase 1 dan juga Anaphase in mitosis, centromere of the chromosome does not split. So that's why kalau awak tengok dekat sini, sister chromatid remain attached. Yang akan terpisah adalah homologous chromosome. Okay, so that's why here it explain each chromosome still has two chromatid attached at their centromere. Okay, so awak kena ingatlah how to draw the structure of the cell that undergo Anaphase 1 correctly. Okay, so you have to draw the chromosome like this lah. Alright, and then next we proceed to Telophase One. Okay, so what happened during Telophase 1? So if you look at here, chromosome reach the respective poles like this. Okay, and then each chromosome is composed of two sister chromatids. One or both chromatids include region of non-sister chromatid DNA. Okay, so if you look at the structure of the chromosome here, okay, so the appearance of the chromosome is composed of two sister chromatid. Okay, maksudnya kita boleh anggap dekat sini, okay, centromere does not split, so that's why, okay, chromosome is in form of sister chromatid. Okay, and then if you look at the sister chromatid here, it has one or both chromatid include region of non-sister chromatid DNA. Kita akan nampak tempat di mana berlaku crossing over tadi, okay, akan ada region lah of the non-sister chromatid DNA. Maksudnya kalau untuk red color chromosome here, okay, it will get the DNA portion ataupun gene portion daripada the blue color chromosome here, so as well as this one. And then what happened to the chromosome? Chromosome uncoil and lengthen. Okay, and then the nuclear envelope and nucleolus reappear. So if you look at this one, okay, akan terhasilnya semula, okay, ataupun terbentuknya semula nuclear envelope and also nucleolus. Okay, And then cytokinesis occur to separate the cytoplasm and resulting to daughter cell. So right after telophase 1 is already complete, so the process is continue with cytokinesis. So what happened after cytokinesis is occur? So this cell will be separated or it will be divided into two daughter cell. Okay, so kalau awak tengok based on the telophase 1 here, so remember, okay, hanya nuklear saja ataupun kita kata nukleoli sahaja yang akan membentuk two daughter nuclei, tetapi masa telophase 1 the cell does not separate yet. Cell hanya akan divide sebaik sahaja berlaku cytokinesis. So means that at the end of the meiosis 1, okay, so two daughter cell will be produced, which the number of the chromosome is already reduced to half. Pada asalnya kita ada 2n equal to 4. Okay, bila berlakunya meiosis 1, so the daughter cell produced will become n equal to 2. Okay.